Methods of preservation of bacterial culture. Introduction. To maintain isolated pure cultures for extended period in a viable condition without any genetic change is referred as preservation. During preservation, most important factor is to stop the microbial growth or at least lower the growth rate. Due to this, toxic chemicals are not accumulated and hence viability of the organisms is not affected. The less number of generation reduces the mutation frequency. Objectives of preservation. To maintain isolated pure cultures for extended period in a viable condition, to avoid contamination, to restrict genetical change, that is mutation. Necessities. So it is used for academic purposes, research purposes, fermentation industry and biotechnological field. Methods of preservation. So first is continuous metabolism. Periodic transfer to fresh media, which is called subculturing. Storage in sterile soil, saline suspension or an oil overlay. First is suspended metabolism. So storage at low temperature, lyophilization of freeze drying, storage in silica gel. So here we do periodic transfer of the organism into fresh cultures. So these are frequently transferred to fresh media once the media are prepared. So every now and then, for at least once in 4 weeks to 6 weeks, they are transferred to new media and they are preserved. Advantages. It is a simple method and no apparatus is required. Easy to recover the culture. The disadvantages are risk of contamination is high and it might be possible to change the genetic and biochemical characteristics. Storage at low temperature or freezing. Freezing is a good way to store bacteria. Freezers can split into 3 categories. First is laboratory, ultra flow and cryogenic. The advantages are it may be used in the successful isolation of many species and cells are viable for around 10 to 30 years without changing their characteristics. So long-term preservation can be done. Next is storage in sterile soil. This method is mainly applied for preservation of sporulative microorganisms, example Bacillus, Streptomyces, Aspergillus and Clostridium. Oil overlay method. This is a similar method which is used for the periodic transfer of organisms. So the only difference here is the use of paraffin oil. Many bacterial species can be preserved by growing the organism on agar slant with a sterile mineral oil or liquid paraffin. Oil must cover the slant completely. Oil reduces the loss of water and slows down the exchange of gases within the organism and its surroundings. So hence the organism can be preserved for a long time. Advantages. It is cost effective and the cultures of bacteria and fungi can be stored around 10 to 20 years. It is highly recommended for anaerobic organisms. The disadvantages are these organisms lose their sporulating ability. Genetic and biochemical characteristics of the microbes changes during periodic transfer of cultures. Next is lyophilization or freeze drying. A process used for preserving biological material by removing the water from the sample, which involves first freezing the sample and then drying it under vacuum at a very low temperature. Freeze drying can be preserved different types of microorganisms that would be killed by ordinary drying. A dense cell suspension is placed in a small vial and frozen at around -60 to -78 degrees Celsius. So this is the apparatus which we use for freeze drying. When I was a student, my HOD never used to let me touch this. The vials are immediately connected to high vacuum. The ice present in the frozen suspension evaporates during this process under vacuum. The results are the production of dehydrated bacteria with a minimum damage to the delicate cell structures. The vials are then sealed under vacuum and stored in a refrigerator. So the advantages are the cultures are viable for more than 30 years. Subculturing is not required. Maintaining the culture without contamination is possible here. Genetic stability and a minimal storage space is required. Transportation is easy. You can put it in a pocket and you can take it anywhere. Thank you.